Very warm good morning to one and all, and a very warm good afternoon to our resource person for the day, Dr. MT Jeena, who's joining us from Korea. So it's morning over here, but on the side of the resource person, it's the post-lunch time, I think. So we gathered today for another session of EXPLORIGATE, the research webinar series organized by the IQAC and the Research and Consultancy Cell of Vimala College, Thrissur, Kerala, India. And EXPLORIGATE for the past 1 week has been having a great run. We've been exploring and investigating research prospects and other concerns related to research and development across disciplines. And from the side of the IQAC and the Research and Consultancy Cell, it has been heartening and overwhelming to see the response and the feedback that we've been receiving so far. We're looking forward to yet another fruitful session in the discipline of science. And today, joining us is Dr. MT Jeena, postdoctoral researcher, Department of Chemistry, School of Natural Science, UNIST, Ulsan from the Republic of Korea. And Dr. Jeena will be presenting a session titled Towards the Development of Promising Anti-Cancer Agents, a Drug-Free Intracellular Assembly Approach. I believe this is an area that is going to be much looked up to, considering the gravity of the topic and the area of research. And with a word of very warm welcome to our respected principal, to Dr. Minimal, IQAC coordinator, the IQAC core team members, and the Research and Consultancy Cell. May I invite Dr. Bijoy P. Matthew, Assistant Professor, Department of Chemistry, to welcome the gathering and introduce the resource person for the day. Over to you, Bijoy, sir.
Thank you, Ms. Maya. Good morning, everyone. Before introducing the speaker, I would like to congratulate this joint effort of the IQAC and the Research and Consultancy Division of our Vimala College. I would like to thank our principal, Dr. Sister Beena Jose, and our vice principal, Dr. Minimor K. Jose, for their constant support and encouragement, not only to me, but everyone behind the successful organization of EXPLORIGATE webinar series. Now, speaking of the resource person for our today's session, Dr. MT Jeena. She is currently the postdoc in the Department of Chemistry, Ulsan National Institute of Science and Technology, Republic of Korea, working under Professor Ja Hyung Ryu. Dr. Jeena, born and brought up in Kerala, she received her bachelor's degree in chemistry from Payyanur College affiliated to Kannur University and master's from Cochin University of Science and Technology, both with university ranks. Dr. Jeena worked for a few months in IISc Bangalore and in National Chemical Laboratory, Pune. Her research about the development of novel anti-cancer medicine received a doctoral degree in 2019 from UNIST, Republic of Korea. In her academic career, the major credentials received are as follows. Dr. Jeena, with her advisor, introduced a new concept of organally localized self-assembly of amphiphilic peptides as a potential anti-cancer strategy and published the research outcome in the Nature journal and obtained US patent for their discovery in 2017. She has published 12 research articles in refereed international journals, 1 US patent, 1 book chapter, and 5 Korean patents so far. Talking about her research journal, she has published in the reputed Nature journal, and also she has a publication in the reputed ACS journals as well. Personally speaking, she is my good friend and a good person apart from her research extravaganza. From my experience, she is a disciplined researcher and an expert in her field. I believe the audience is going to be influenced a great deal from today's session. Now, with these words, I would like to now give the stage to our speaker, Dr. MT Jeena.
So good morning to all. Can you hear me? Is it okay? Sound is okay?
Yes, yes.
Yes, ma'am. Yes, ma'am.
Okay, good morning to all, and thank you for inviting me for this talk. And I know that Kerala people or Keralites are going through very difficult situation because of yesterday's plane crash and Corona. So I think for a while, let's keep all those things apart and let's have some scientific talk for about 1 hour. So shall I begin my talk? Okay. So as introduced, my name is Jeena and I am postdoctoral researcher at Department of Chemistry, UNIST, and it is in Republic of Korea. So Korea, let me first introduce Korea. So Korea is a small country which is situated in the Eastern Asia. So Korea is here as a small country. So Kerala is here. It will take about 16-hour flight from Kerala to Korea. So whenever we speak about Korea, we have a very wrong concept because we think that it is the land of the terrific leader Kim Jong-un, and the situation in Korea is very dangerous. So situation is not like that in reality. Korea is actually two. There is South Korea and the North Korea. North is actually the Kim's land, and South Korea is entirely different from North. I would say it is one of the most beautiful place in the world. It is a small country of about only 52 million population, and life in Korea is quite heavenly because it is quite technologically advanced country, and most of the world renowned companies such as LG, Samsung or Hyundai, that is from Korea, which means that they are technologically quite developed and rich country, and they have a beautiful landscape. And in case of food, I think Korean peoples usually eat very healthy food. They don't use many oils or spices. And in case of city life, it is very technologically advanced and well organized. So life in Korea is kind of living in a heaven. So I really recommend, if you have a chance to visit Korea, don't miss it. It will be a great experience for everyone. Okay. So in Korea, there are many universities such as KST or Korea University, GIST or POSTEC. So all the universities in Korea is highly devoted to research. They have all the high-tech technologies. So research in Korea is a unique experience because everything is faster, making data or developing some new concept and establishing a new research. Everything, Koreans are so fast in that. So doing a research in Korea will be always a unique experience. And my university is UNIST. I have done my PhD in UNIST. It is called Ulsan National Institute of Science and Technology. So this is the whole picture of my university. So it is established in 2007, and the goal of our university is to be within top 10 by 2030. So which means that they are highly devoted to research. They have very heavily invested laboratories, and they produce high impact publications every year, and it is still have a top ranking. And what make a research in UNIST unique is that we have all the facilities that is necessary for the research. Doesn't matter it is a chemistry or biology or physics or engineering, we have the central facility and we can always use all the equipment ourselves and get data within no time. So this is why the research in UNIST is a unique experience. So if anybody wanted to go for higher studies abroad, I recommend this place strongly because it will be a great experience and will contribute greatly towards our career and the academic contributions. So moving towards my research, the topic for my research is towards the development of promising anti-cancer agents, a drug-free intracellular assembly approach. So if I explain my topic in detail, I'm going to talk about the development of anti-cancer agent, which means that certain agent that can act against cancer. And it is a drug-free approach, meaning we are not using any conventional drug, but free of drugs. But we use a new concept called intracellular assembly, which means that make a self-assembly inside of a cell. So that is what I'm going to talk about. And we have to familiar with all the terms before we move into the research deeply. So self-assembly, what is mean by a self-assembly? Self-assembly is self-organization of certain building unit into nanostructures, into a well-defined nanostructures. This process is called self-assembly. So self-assembled structure may be a micelle or a fiber-like a structure or a kind of ribbon-like structure. Okay, so this process is called a self-assembly. So what could be this building unit? There are several candidates for the building units, such as peptides or polymers or small molecule. And here I am going to talk about the anti-cancer agent. Okay, we are going to develop anti-cancer agent from this concept. So whenever we are talking about the biological applications, the peptides are always the best candidates because they are highly biocompatible, they are low molecular weight, and it is easy to produce, and size and structure we can vary. So peptides, we use this peptide building unit in order to make the self-assembly in this talk. Okay, so what are the peptides? Peptides are made from amino acid. We know that there are 20 natural amino acids. Amino acids are the compound which consist of both the carboxylic acid group and the amine group. Okay, more than 2 amino acid can couple to produce the peptides. So these peptides that consist amino acid as the building unit, which can serve as the perfect example for the self-assembly. So peptide can self-organize to form the self-assembled structure. So we are going to use the peptide in this application. So as I explained, the self-assembly means self-organization of building units, let's say peptides, into the nanostructure, self-assembled nanostructure. So we are going to make this process inside of a live cancer cell. So before we are moving towards deeper into this talk, we have to be familiar about what is a cell. So whole of my talk is about the cell. So we should know what is cell, and we should have a basic idea about the cell. The cell is the basic building unit of all living system. Okay, this is a schematic representation of a cell. Cell we can represent like that. It have a cell wall called a plasma membrane, and have a nucleus where our DNA or genes are stored, and have the endoplasmic reticulum and the mitochondria and several thousands of other components. So cell structure is about 20 micrometer. Okay, so we are going to do this self-assembly process, that is self-organization of building unit or peptides into nanostructure inside the cell. And this is the microscopic image of a live cancer cell. So the cell looks like this. It have about a 20 micrometer size, and you can see the nucleus here. So we have to make this process inside the cell. For what? In order for them to die, or in order to do a process called apoptosis. Apoptosis means cell death. The process of cell death is called apoptosis. So we will do this self-assembly inside of a cell in order to kill it. And this is the microscopic image of the cell that undergone apoptosis. This is a live cell. If the cell die, it looks like that. So this is whole basic idea about the cell. And we are going to do a cancer therapy using this concept. So we should know what is cancer and what are the treatment options at present, and why we need a new approach for a cancer therapy. So cancer is a disease that is characterized by uncontrolled growth of the cells. Okay, so I mentioned that cell is the basic building unit of all the living system. We are all made up of trillions of cells. So how this process is maintained? A cell continuously undergo a process called cell division. So this process always occur in our body, and when the cells are getting damaged, they are removed from the body themselves by a process called apoptosis. So whenever I say apoptosis, it means that cell death. I will use this term many times, so please be sure apoptosis means the process of cell death. So the cell continuously undergo division, and when they are getting damaged, they are removed from the body by the process called apoptosis. And when the cells are getting older, then also they will be removed from the body by the process called apoptosis. And this is a cyclic process, and this continuously happen in our body. So on one hand, new cells are produced by cell division. On another hand, cells are damaging and getting older, so they are removed by the process called apoptosis. Okay, but what happened in the case of cancer? In the case of cancer, what happened is that this process of cell death or the apoptosis, that is prohibited somehow due to some mutation or due to some structural change or the functional change in the cell. The cells are no more getting died, but they produce continuously without damage. As a result, what happened? As a result, there will be a continuous accumulation of this cell. New cells are producing, also the older cells are not removing from the body when they should be removed. So there is a uncontrolled growth or uncontrolled proliferation of the cell, which is mutated. The mutation means it can be a mutation in the apoptosis related protein, or it can be a mutation in our DNA. Okay, and this condition is called a cancer. So cancer is basically the accumulation of the unwanted cell or an uncontrolled growth of the cell. So what we do for a cancer treatment, we have to remove these uncontrolled cells from our body in order to treat a cancer. So this is whole the basic idea about cancer. So cancer is an uncontrolled growth of the cell. And there are four different stages for a cancer development. There are basically four stages. As I explained, it starts with a mutation in a single cell. Okay. At that stage, when a cell is mutated, we don't know that we are going to develop a cancer or we will have a cancer. Okay. And this mutated cell later grow. They started growing and they started proliferating. And in the stage one, it become more in numbers. Started with a single cell, now they are multiplied and have an enlarged number, more of such cells. In this stage one, we can detect the cancer. It is microscopically visible. We can understand that we are going to develop a cancer. In the stage two, they continue their proliferation and they are more and more, and it is enough grown to felt as a lump. This is stage 2 cancer, and in the stage 3 they are spread from the initial state to the surroundings. This is called stage 3 cancer. And stage 4, it is called metastasis, which is the most dangerous stage of the cancer. If a patient develop the stage 4 cancer, then it become very difficult to treat. Almost the survival rate is very poor if a person reaches stage 4 or a metastatic stage. So metastasis means it is just first initially start with a single cell, now spread to the surrounding. Now it is spread to other part of the body rather than their original state. So this is called metastasis. Metastasis basically means spread of cancer from one part of the body to other part of the body. And why this happen? Suppose we have a cancer in the bone, but in the metastatic stage, we will have a cancer also in the lungs, because they are carried by the blood flow from the original state to the another site. So this is called metastasis. And depending upon their site of occurrence, whether it is in the throat or in the lung or stomach, they are given different name. Basically, the mechanism is same. This start with a mutation in the single cell and the uncontrolled proliferation. So all the cancer occur both in male as well as in the female, but two cancers are specific for women, that is ovarian cancer and the breast cancer. And why should we develop this cancer and what are their cause? It starts with a mutation in a single cell, and it can be caused by some hereditary factors or exposure to the UV rays or some chemicals or smoking. Smoking is one of the biggest reason for the cancer development, and also some virus can also induce this type of mutation. And apart from this, our lifestyle or fast food intake, all of this can cause cancer nowadays. And how we can treat this type of cancer? There are still different options in order to treat the cancer. So this ribbon symbol is for the cancer. Each color represent each cancer. For example, this blue color represent colon cancer, and the yellow color represent a child cancer, and this kind of violet color, it represent all cancer. So it is combination of all cancer represented by this violet color. So if you Google it, we can see what represent each color. So there are different options in order to treat the cancer. So one is called chemotherapy. For the chemotherapy, we uses a chemical drug to kill the cancer. Rather than that, we can use a surgery. We can use a surgery in order to remove the cancer, or a photodynamic therapy, or a radiation therapy, or an immunotherapy. So what's happened in each method? In case of surgery, we remove the tumor by surgery. By the operation, we operate and remove the tumor. But problem of surgery is that even though we remove surgically the tumor, there can be traces of cancer still remaining. It can again cause the reoccurrence. even though patient done a surgery for the tumor, maybe later they can again develop. In that case, we have to do a chemotherapy after the surgery. And photodynamic therapy use irradiation of light on the photosensitizer, and that photosensitizer will produce toxic reactive oxygen species and remove the cancer. And in case of immunotherapy, we use body's immune power to fight against cancer. But immunotherapy alone cannot work. They should be always in combination with the chemotherapy or a radiation therapy. In case of radiation therapy, we use high energy X-rays in order to kill the cancer cell. So of the all available methods for the cancer treatment, chemotherapy is considered to be the best way and the most promising way, because it makes sure the complete elimination of the cancer. It is less costly and reduce the risk of recurrence. Okay, compared to all other method, this chemotherapy is the one which can make sure the cancer is completely cured. So many of the research is focused on chemotherapy in order to improve the efficiency of the available chemotherapeutic agents. Okay. And chemotherapy is, as I explained, chemotherapy uses a chemical drug in order to remove rapidly growing cell, that is cancerous cell. There are different mode of administration for the chemotherapy. It can be either taken by oral medication, like in the form of a tablet, like we do for the other diseases, or it can also be injected directly into our body under the skin or into the muscle or into the vein. These are the different methods how a chemotherapy is performed. So starting from 1940, there are more than 150 FDA approved anti-cancer drug available now in the market. FDA means Food and Drug Administration. So all the drugs need their approval in order to be available in the market. So it's the first drug is approved by FDA in 1949. Then it is for the lung cancer. It is called methylchloroethamine, have this type of structure. So all these available 150 drug belongs to two categories. They are either belongs to a cytotoxic drug or targeted drug. So there are two type of drugs for the chemotherapy available now in the market: cytotoxic and targeted drug. Let's see what are those. Cytotoxic drug means those are the drugs that can directly damage the DNA of a cancer cell. So we know that where is DNA is located? DNA is located in the nucleus. So cytotoxic drug, what they do is that they destroy the function of DNA. We know DNA means it is the very basic. DNA means they have the double strand guanine and cytosine, adenine and thymine. They should have a base pairing in order to function properly. So what is cytotoxic drug do is that they intercalate between the strands of DNA and cause them to mispair or stop their replication or break down their strand. This is what a cytotoxic drug do for the chemotherapy. And eventually, if the DNA is damaged for the cell or for this cancer cell, then cell will undergo death. And one example of a cytotoxic drug is called Doxorubicin, have this type of structure, and this is now widely using as a chemotherapeutic agent at present in the market. And this is just one example of the hundreds of drugs. And another type of drug is called a targeted drug. What a targeted drug do is that they target some proteins or enzyme that is overexpressed in the cancer cell. So I said that cancer cell is the uncontrolled proliferation of the cell. For them to uncontrollably proliferate, there are some factors which is overexpressed in the cancer cell compared to the normal cell. So a targeted drug targets those factors. It could be an enzyme or some growth factor or some hormone within the cancer cell. And the targeted drug bind with such factors or such as enzyme or protein, and then inhibit their function and eventually prohibit their growth. And one example of one such a drug is gefitinib. It is an EGFR inhibitor. EGFR is a growth factor. So all of the available chemotherapeutic drug at present belongs to two categories, either cytotoxic drug and targeted drug. Then why we need a research? Anybody can think we have already a 150 drugs. Then why we need to further research about this? The reason is that if we see the cancer-caused death every year. There is millions of people die every year by different type of cancer, which means that the available chemotherapy or the available treatment for the cancer is not at all effective. So we need to improve the treatment strategy in order to avoid this situation. That is, in order to avoid a cancer-caused death, we need to improve the treatment. We need to develop more efficient chemotherapeutic agent. So that is why we do the research in this area. So why this happen? Why the chemotherapy at present is not effective? That is because there are certain drawbacks associated with this conventional drug, or there are some challenges associated with the cancer cells that we should overcome. The first things comes about the selectivity problem. Even though there is a more than 150 anti-cancer drug, they are not enough selective to the cancer cell, which means that we have the cancer only on a special location. We don't have cancer in all our body. We have only in special location, like maybe in stomach or lung. And we have to do a chemotherapy in order to target only those specific location. But in reality, what happens is that when we take a chemotherapeutic agent or when we take a cancer drug, it may be destroy all the cancer cell, but along with that, it may also destroy the normal cell. It may hamper our healthy life. Okay, and this is why we see certain side effect while doing the chemo. So we may be seen, some of you may be seen in real life, or most of us seen in the movie, that after doing a chemotherapy or patient affected by the cancer have the severe hair loss. So that happens because the chemotherapeutic agent also affect our normal cell. That is why we have the side effect. Not just a hair loss like a fatigue or nausea or vomiting tendency. There is severe side effect associated with the present chemotherapeutic agent. So this is the first drawback, or the important drawback. It arises from the selectivity. So we should develop some anti-cancer agent that can only act in the special location, or only act in the cancer cells. This is what we have to overcome at present. And second one is drug resistance, which means that certain drugs are highly resistant towards the therapy. So what is mean by resistance? Resistance means they are not responsive towards the drug. They have high power to fight against the drug we are taking. There are two types of resistance. One is called primary resistance and another is called acquired resistance. Primary resistance means in case of tumor, I said it is an accumulated cell. Okay, so there is many cells. There can be two type of cells in a single tumor. One is cells which are highly sensitive to the drug. Another is more powerful. They are not affected by the drug. They are called resistant cell. So what happens in such a case? Even though we take a drug, only the sensitive cells are died. But resistant cell, maybe they are small in number, but they can again rapidly regrow and develop the tumor again. So what happened? What is the end result? Even though we take the drug continuously, the cells are not affected. Okay, so the tumor still, in the end, tumor still is as such. That happens because of their resistance towards the drug. And another one is called acquired resistance, which means that the cells acquire a resistance due to the drug. Initially, the cells may be sensitive towards the drug. They may be highly responsive towards the drug. But what happens when we take a drug, or when we take the drug usually affect their drugs are usually small molecule. They usually act on the gene or the DNA or some proteins or enzyme that are present in the cell. In some cases, they may inhibit their function effectively. However, in some cases, they can just do some interaction. The drugs are quite small and proteins are quite huge in terms of molecular weight. So they may be not able to inhibit properly. Instead, the drug and protein or drug and gene just interact each other. As a result, the protein will develop a mutation. And this mutated cell again regrow and produce more and more mutated cell. But drugs are designed in such a way that they can recognize the protein or gene only in their original state, but not in the mutated state. In that stage, we say that cells acquire a resistance towards the therapy. So what happens when we take a drug? Initially, the cells are sensitive, but some cells eventually develop certain resistance, and those resistant cells again regrow as a tumor. But even those normal, even though the sensitive cells are die, so end result is that therapy become ineffective. So this is the second problem associated with the present chemotherapy, that is drug resistance. And third problem is tumoral heterogeneity. So heterogeneity, the term indicate hetero means different. So what is tumoral heterogeneity? Which means that there can be cells which is different. So there are two type of tumor. It may be homogeneous and heterogeneous. Homogeneous tumor means all the cells are similar. So if we take a drug, it may be act on all the cells, but most of the cases, tumor is heterogeneous in condition, which means cell may be different type. So different type means there may be some protein expressions are changed or enzyme expressions are changed. So there can be different population in a single tumor. So when we take a drug, only some small portion of the cells are died, but some can survive, some are not responsive towards the drug. Such survived cells can again regrow and again repopulate, and again the tumor can develop. So these are the three major problems associated with the present chemotherapy: selectivity, drug resistance, and tumoral heterogeneity. So in order to solve this problem, in order to overcome these problems, there are thousands of research going worldwide in many cancer research laboratories. So the aim is we have to improve the efficiency of chemotherapy. So mostly what people do is that they develop certain vehicle in order to encapsulate the drug or in order to deliver the drug properly into the tumor. But this research may be successful in terms of selectivity. They may be able to deliver specifically into the cancer cell. But the problem associated with the drugs, such as drug resistance or the tumoral heterogeneity, these are still need to be solved. In order to solve this problem, scientists recently introduced a new method that is called self-assembly inside of the cell for cancer therapy. Okay, so in here what we do is that there are 150 anti-cancer drugs. We are not using any of this conventional drug. Instead, we design some molecule which can form the self-assembly. So some building unit, usually from the peptide, which have the intrinsic ability to form the self-assembly. So speciality of this type of building unit is that they have no activity when they are in the molecular state. They are not responsive towards anything. They are kind of inert. But what is the case of a conventional drug? Conventional drugs are also small molecule, but they are highly toxic in the state how they are. They can cause toxicity in the molecular state, even though they are small molecule. That is why we have the side effect, and that is why the normal cells are getting affected. However, in case of this type of approach, we design entirely new building unit that have no activity. However, they can enter inside the cancer cell and do a self-assembly process inside of the cell, usually self-assembly to form certain nanostructure and usually the fibers. So, and then they can turn out the activity only inside of a cancer cell, but nowhere else. And this is a quite recently introduced method in order to kill the cancer cell effectively. And the advantage of, so why we do this one? We do self-assembly inside of the cell in order to induce apoptosis or cell death. So what are the advantages of this method? They are highly selective to the cancer cell because we design the building unit in such a way that they can go specifically inside the cancer cell and do self-assembly only in the cancer cell. And there is no acquired resistance, because acquired resistance comes when the drug interacts with some proteins or some enzymes or gene. Here the self-assembled structure is not interacting with any proteins or gene. The self-assembled structure induces a toxicity, or self-assembled structure kill the cell. So there is no acquired resistance, and no matter it can work in the tumoral heterogeneity condition, no matter how differ each cells, even though they have the heterogeneous condition, this building unit only need a cell in order to make the self-assembly. Only need a cancer cell in order to make the self-assembly. So they can work under the tumoral heterogenic condition, and it will work even for the drug resistant cells, because as I explained, even though the Normally the drugs may be resistant towards the cancer cell, but in here the toxicity is induced by the self-assembled structure, so there will be no risk of acquired resistance, and it can also work for the drug resistant cell. And how this concept is derived, and this is derived from the neurodegenerative disease concept. We know that we may be familiar about the disease such as Alzheimer's disease or Parkinson's disease. Alzheimer's disease, it is the loss of memory. So maybe one example, maybe if you remember the movie Tanmatra, so in that the Mohanlal get affected by the disease called Alzheimer's, loss of memory. So why this one happened, or what is the reason for such a memory loss? This happens because of a specific protein called amyloid protein. So we all have the amyloid protein. However, in some cases, usually in the older people, this amyloid protein undergoes some mutation and develops some fibrils. So they aggregate and make some fibrils. These fibrils are the one which is toxic, and they eventually cause the Alzheimer's disease or many other neurodegenerative disease. So scientists borrow this concept. So here the amyloid fibrils induce toxicity and cause the Alzheimer's disease, which means that these fibrils can induce the toxicity. So scientists, cancer researchers borrow this concept, and they get an idea that the fibrils, if we induce this type of fibrils inside of a cell, then they can induce the toxicity towards the cancer cell also. So that is why we are interested in the making fibers inside of the cell in order to induce specific cell death for the cancer cell. And how this strategy really work? If we see the cell, as I explained in the initial slide, the cell consists of many components, several cellular organelles such as nucleus, mitochondria, or endoplasmic reticulum. Along with that, there are also several fiber-like components which maintain the structure of the cell, such as beta-actin or vimentin or tubulin, more specifically microtubulin or cytoskeleton network. All the cell have this kind of structure, and basically all these structures are fibers, is a kind of fiber. So cells already have a many type of fiber. So what will happen if we introduce a new fiber inside the cell? We are introducing a synthetic fiber inside the cell, okay, or we are making an extra fiber inside the cell. When we make such a fiber inside the cell, they can interact with the fiber-like a structure that is already present in the cell, maybe tubulin or vimentin or beta-actin or cytoskeleton. Then eventually, if they interact each other, then they are destroying the cell's harmony, and they will eventually cause the release of apoptotic bodies and induce cell death by the process called apoptosis. So I said that the self-assembly is the process of spontaneous organization of the building unit into the nanostructure. So for a building unit, in order to make a self-assembly, they have to interact each other. So how this self-assembly process happens? So the building unit interact each other by various non-covalent interactions such as hydrogen bonding or pi-pi stacking or hydrophobic interaction. So they have to interact each other. They have to come closer in order to organize themselves into nanostructure. So how this is not, it is not like some building unit immediately make a self-assembly. They should have a certain forces like this. For that, they should have a certain concentration called a critical aggregation concentration. So there should be enough number of such a molecule in order to make this type of assembly. So that particular concentration, when such a building unit make a self-assembly, is called a critical aggregation concentration, or in short CAC. So above this CAC or above this critical aggregation concentration, the building unit which have the self-assembling propensity will exist as a nanostructured form or self-assembled form, and below this concentration they will exist as a small molecule. So this is very important for the self-assembly. Self-assembly is a function of concentration. Only above certain concentration, they are self-assembled structure, and below that concentration, they are still molecule. And inside the cell also, the molecule have to overcome this CAC. It is not like we make some molecule inside the cell and they immediately undergo self-assembly. No, it is not the case. There should be enough concentration in order for them to interact each other and make a self-assembled structure inside of the cell. So if we consider the cell size, it is about a 20 micrometer. So the human cell have a size of 20 micrometer. So this building unit have to disperse in the whole 20 micrometer space and interact each other and make a self-assembly structure. And this is a little bit difficult task because our goal is that we are making some concept and we are developing new drug in order to commercialize it. So in order to make them a self-assembly inside this huge area, they need to be high concentration. That is not a good sign because drug should work in the lowest possible concentration. We have to minimize the effective concentration of such drugs. Okay, it should be active at lowest possible concentration. But when we consider the whole cell, because of their size is a 20 micrometer, it's always quite huge. The concentration required is in the millimolar range. The molecule need to be in several millimole. This is not good because we don't want to produce such a large scale. We should minimize their concentration. So what can we do for that in order to minimize the effective concentration of a drug? Instead of making the self-assembly inside the whole cell, we decided to equip the self-assembling unit with some targeting ligand, so that instead of making assembly in the whole cell, now they can target a specific cellular compartment. Okay, so initially we thought we can make an assembly in the whole cell, but that have a drawback of concentration problem. So we had to minimize the concentration to the lowest possible. So we decided to equip this self-assembling unit with a targeting moiety, which means that we can target the cell as well as we can target a specific subcellular compartment such as mitochondria. So we know that there is various component in the cell. So mitochondria is one of the important component. So we equip the self-assembling unit with a mitochondria targeting ligand. So now the building unit, instead of assembling in the whole cell, they just target the mitochondria or in the nucleus or in the endoplasmic reticulum. So basically, we target a small subcellular location of the cell. As a result, what will happen? The molecule can make a self-assembly inside the mitochondria instead of whole the cell, called an intramitochondrial assembly, or in the nucleus, called an intranucleus assembly, or in an endoplasmic reticulum, called an endoplasmic reticulum assembly. So now the summary is that instead of making the self-assembly in the whole cell, we are making the self-assembly of such a building unit in the small subcellular compartment, like a mitochondria or nucleus or endoplasmic reticulum or any other organelle of our interest. So in general, we can call this one as an intra-organelle assembly or an organelle localization induced self-assembly, or in short, it is called as an ELISA strategy. Okay, so which means that we make a self-assembly in the small subcellular compartment in order to kill the cancer cell. So how this strategy work? This is quite simple. We make some molecule which can enter inside the cell and target a certain subcellular compartment. So they accumulate in a small area of the cell. So now what is happening here? The molecule that we designed initially in the outer cellular space, they have a very small concentration. Okay, so from the outer cellular space, they enter inside the cell, and then they again enter in this small subcellular compartment. As when the whole molecule are collected in a small space, small subcellular compartment, they experience a much higher concentration than their actual concentration. So the effective concentration that molecule suffer is very high compared to the actual concentration, which means that such a molecule can immediately overcome their CAC and interact each other to form the self-assembled nanostructure and thereby kill the cell. So usually this nanostructure kill the cell by destroying the membrane. destroying the membrane of the organelle. So when the organelle's membrane is destroyed, every enzymes, protein or apoptotic bodies stored in the membrane is released to the cytoplasm, and eventually they will cause the apoptosis, or eventually they will induce the cell death. So this is how the whole concept works. So we design some organelle targeting moiety, and it will target certain organelle and do the self-assembly inside the cellular organelle and kill the cells. So I think this is too much about a new concept. So I would like to hear your doubt, and let's clear the doubt and move towards the next topic. So if you have any doubt, please feel free to ask. Do you have anybody?
Ma'am, in the chat box, one Miss Anila Antonia has asked, can we use shape memory biopolymers for cancer cell treatment?
Can we use shape memory biopolymers for the cancer? Actually, we can, but what kind of biopolymer is important? What is the structure of polymers, or what is mean by polymers? Are a huge area, and there is different type of polymers, right? So of course we can use the polymers for the cancer treatment, but it is important what is the structure and what is their molecular weight and how they function. Everything is important. Of course we can use, but thing is that we have to design it properly, and we should be clear about how they will function, in which what could be, if we want to use for the cancer treatment, we should induce the cell death properly. So we have to be clear about that. It is important to know the structure of that. Of course, there are so many polymers example that we are using for the cancer treatment, and we can use it. How liposomes effective in transfer of alkaloid of a plant to cancer cell? Actually, I think this is the question need to be little more specific. The liposomes effective in transfer of alkaloid of a plant to the cancer cell. I think I'm not clear about that question. So somebody asked my email ID. I will put my email ID here, so you can ask.
All right. Ms. Pinky Shebin, you could also unmute and clarify if you want to ask further. Thank you, Jeena ma'am, for sharing your email ID. Yes, you're audible. Please speak up. Yes.
Ma'am, this plant is having a lot of alkaloid compounds. These alkaloids are used as an anti-cancer agent. But the dose of alkaloid is very critical, because if we exceed the dose of alkaloids, it can cause toxic nature. So we need to carry these alkaloids from plants in a minimum amount to the targeted cell. How this is possible? How the liposome coating helps in the transfer of alkaloids of the plant to the target cell?
Actually, this is not a simple process. The question, so basically you ask how you can use alkaloid for the cancer treatment. That is what you wanted to know, right?
Yes.
So how we do a cancer treatment? Actually, it should be start from the very basic. The first step we do is that we culture the cell lines by ourselves in the laboratory and test the toxicity. Okay, if you think some alkaloid have an anti-cancer property, so we have to first test them in the laboratory, and we have to find the toxicity range. Okay, at what concentration you think, for example, 1 micromole, right? So we have to do a cell viability assay for the cancer cell at different concentrations.
In vitro test. Okay.
In vitro, and then we have to find what is the concentration that molecule is toxic to the cancer cell. Okay, at the same time we have to also test them in the normal cell. Most of the toxic agent that is toxic to the cancer cell may be also toxic to the normal cell. This is the first step. We have to make sure your alkaloid is only toxic to the cancer cell, but not to the normal cell. Then only we can go forward. Okay. So the first is you have to find the range of your toxicity.
Yes.
Okay.
You're right.
Okay. Then, have you conducted clinical trial using self-assembled molecule from Sina Jojo? Actually, we are going towards the clinical trial. This strategy is introduced in 2017. As you know, in order to go towards the clinical trial, we had to pass many more steps again. So we are developing further, and we are continuously doing the research in order to improve the in vivo efficiency and such things. So we will be doing a clinical trial in future, but we have just started in 1 year back. So we will definitely be going towards it. So we are doing more and more research on this to bring this strategy to the clinical trial. And another question. Okay, beautifully explained. Thank you. That is an inspiration for me to go further. Okay, do you have any other question? Or I think you can ask the further question in the next, at the end of the session. So now I think I will continue my talk. So now can you see the slides? Could you please say? Hello, can you see
Yes,
my
ma'am. Yes, ma'am. Yes, ma'am. Your slides are clear.
Okay, so now until now I have explained what is the mechanism. Okay, we have stopped here. I have explained how this molecule work. Now we are moving towards the real research, what I have already done in the laboratory. Okay, so I said that this is a mitochondria, this is an organelle localization induced strategy. So first step, we have to choose an organelle of our interest, and we have chosen mitochondria for our first trial. Okay, as I said, there can be many organs such as mitochondria, nucleus, or lysosome. There is many options, but first we choose mitochondria. We know that mitochondria is cell's most important component because it is where the energy is produced. Okay, the energy that is needed for our whole life, that is produced from the mitochondria. So if mitochondria is a small organelle of about 500 to 1000 nanometer, and it have an inner membrane and an outer membrane and a matrix. This is how a mitochondria looks like. And why we choose this mitochondria for our further studies? That is because I said that normally our cells undergo cell division, right? The cell had to be divided, and then this process had to be continued, and the cell had to be die also. This is a cycle, cell division and the cell cycle. But what happened in the case of cancer cell, as I explained, cell is not dying. So there is no cell death or there is no apoptosis. So this process of apoptosis begins from the mitochondria. So mitochondria is the cellular component which begin the apoptosis or the cell death process. So cancer cells are not undergoing cell death, which means that mitochondria of the cancer cell is dysfunctioned, or somehow apoptosis related proteins from the mitochondria is not releasing from the mitochondria to the cytoplasm in order to begin a cell death. So because of that reason, we think that mitochondria could be the best target in order to kill a cancer cell efficiently. And what we are going to do is that we will be destroy this mitochondria of the cancer cell and initiate the process of apoptosis inside the cancer cell and begin the cell death, so that we can remove this cancer cell that initially have no cell death, but now we will induce the cell death by destroying this mitochondria. So how we can target the mitochondria? So very thankfully, mitochondria have a special features that allow the chemistry people or that allow the researchers to target them efficiently. So if we see the structure of mitochondria closer, the mitochondria have a two membrane, an outer membrane and an inner membrane. It is like that. Okay, the outer membrane is the membrane of the mitochondria is negatively charged, and it is hydrophobic. All the membrane of the biological system that are hydrophobic. Okay, so this hydrophobic membrane, and this is negatively charged because of the function in that. So because of this reason, any molecule which have a positive charge and a hydrophobicity can penetrate this mitochondrial membrane and localize inside. So usually the positive charge is needed because they have to cross a membrane which have a negative charge. So mitochondrial membrane is negative charge, means if you use a negative molecule, then they will repel. They cannot penetrate the membrane, and they are hydrophobic. So if you have a hydrophilic unit, then they cannot interact each other. Only hydrophobic and hydrophobic will interact each other. Okay, so any molecule which have a positive charge and a minimum hydrophobicity they can penetrate this mitochondrial membrane and go inside. So usually people use a special compound called a triphenylphosphonium. This have this type of structure. So 3 phenyl group and a phosphorus and a positive charge. So they are delocalized. Okay, there is a highly delocalized positive charge. And this is hydrophobic. Of course, the phenylalanine is hydrophobic. Okay, they have now hydrophobic and positive charge, so they can penetrate the mitochondrial membrane and go inside. So what will happen when we use a triphenylphosphonium, or in short TPP, for targeting mitochondria? They will first enter inside the cell, and from the cell they cross the mitochondrial membrane and accumulate there. So as I explained before, outside extracellular space, the molecule have a very low concentration, but first they are enter in the cytoplasm. In the cytoplasm, they will feel much more higher concentration than the outside. But when they are localized in the mitochondria, they will feel much higher concentration. Initially low, in the cytoplasm it is 5 to 10 times higher. However, in the mitochondria it is 100 to 500 times higher than the actual concentration. So if we use this type of TPP in combination with another compound, we can this R group means it can be anything. We can design the molecule according to what is our purpose. Okay, so this R group, you have to imagine another compound, another molecule here. So that such a molecule designed with the TPP can efficiently penetrate the mitochondrial membrane and localize inside. So we are going to use this strategy in order to prove our study. So this is the compound of our interest. This is named as Mito-FF. So I will explain why the name comes. So this is the molecular structure which consists of TPP here for the mitochondria targeting. So since it can target the mitochondria, we have given the name Mito. And I'm sorry, somebody have a chat message. Let me please check it. Okay. So if possible, please don't do a chat. Please don't drop a chat between the talk because I am getting disturbed. We will have a question and answer section in the end. Okay. So in case of the molecular design, so we explained Mito-FF. Here is a mitochondria targeting unit which can target the mitochondria, and we use phenylalanine as a building unit. So we said we are going to design something with a peptide. Okay, so phenylalanine is an amino acid. So when two phenylalanine join, they become a peptide. So we are using this type peptide or phenylalanine. Why we choose this one? Because they are highly hydrophobic and they have the intrinsic ability to form the nanostructures. It is quite well known for several decades before. This phenylalanine molecule can self-assemble together to form the self-assembled nanostructures such as fiber or nanowire, depending upon what are the other part. So phenylalanine itself can form fiber. However, it can be changed. What is the other moiety that we are conjugating with it? So here we are using a pyrene and a TPP. So pyrene is needed for the fluorescence. So we know that if you are a chemistry student, then we know that pyrene is highly fluorescent, and it have a special property of hexameric emission because they have a hydrophobic interaction and they have a pi stacking. So we are equipping this pyrene for the fluorescent detection because we are introducing a new concept to the world. So we have to show a bunch of data in order to prove that we are successful in that. So for that, we have to visualize them inside the cell and show that this happens really. So we equip this fluorophore here in order for the fluorescent detection. So this whole structure make a molecule called Mito-FF, which have a TPP and pyrene and a phenylalanine. TPP for the mitochondria targeting, phenylalanine for the self-assembly, and pyrene for the fluorescent detection. And we represent this one like this as a cartoon for our ease, because we cannot use this molecule everywhere in the scheme. So this cartoon which shows, this red shows TPP, this one shows the self-assembling unit, and this blue shows the fluorescent, the pyrene. Okay, so this one now can enter inside the cell and target the mitochondria. So this is what we have to prove. So this is what I have proved in my research. So this is the concept. This is a schematic diagram of all concept. Molecule enter inside the cell, enter inside the mitochondria, get accumulate there. During their accumulation, they make a concentration above their CAC, because this is a small space compared to the cell or compared to the extracellular environment. So now they accumulated, rise their concentration above their CAC. So when there is a concentration above their CAC, they immediately undergo self-assembled structure into nanofibers. So these I explained that we want to make a fiber inspired from the amyloid strategy. Okay, so it can form the fiber inside the mitochondria. So what fiber will do with the mitochondria? Fiber will interact with the mitochondrial membrane and destroy the mitochondria, release the apoptotic bodies from the mitochondria to the outside that will induce the cell apoptosis. So this is the whole concept, and we have done several research in that, and we have published our research outcome in 2017 in Nature Communication and 2019 in ACS Nano and 2020 in Chemical Communication. So I will explain how we have proved this strategy really work. So this is the important step when we do a research. We have to start from the very basic. First, we synthesize the molecule. It is quite easy, or the most easiest step. We synthesize the molecule, and first we have analyzed their mass and the HPLC. We have done every characterization. So I am not showing such data because those are just basic data. So we have made sure that this molecule is formed, and we have synthesized this by solid state peptide synthesis. It is a non-protocol. We can synthesize this peptide building unit first, and then we have coupled with the pyrene butyric acid by the HBTU coupling, and then we have conjugated TPP here using the TPP-Br and the amine. The bromine react with the primary amine, okay, to give this product. So this is the structure MitoFF. And after synthesizing this one, we have checked their transmission electron microscopy. That is, until now I only said they form the fiber. So now we have to prove they are really forming the fiber. So we have done their self-assembly studies in the solution, and we drop in the TM grid, and we have observed them by the transmission electron microscopy called TM, and we could see the fiber, narrow fibers of about a 10 nanometer diameter. This is the diameter distribution. So they are quite small diameter. The mitochondrial size is 1 micrometer or 1000 nanometer. So their diameter is 10 nanometers. So they can easily form the fiber inside the mitochondria. And I said the molecule should overcome a concentration called a critical aggregation concentration in order for them to make an assembly. So I have analyzed what is their critical aggregation concentration. So it must be low, because if we have a millimolar range concentration, it is not a good option to use for the biological studies or the cancer therapy. So I want them to have a low concentration. So quite fortunately, they have the CAC of about 60 micromole. That is very low for them to sell. That is very low or very reasonable concentration in order to make an assembly inside the cell. So we have measured this one by a method called a pyrene excitation method. Okay, and then here this is the critical aggregation concentration. This is the transformation point. So below this concentration, they exist as a molecule. Above this concentration, they exist as a fiber. And after that, I have checked the FTIR spectra and see what are the bands. FTIR spectra of this one means FTIR spectra of the fiber. So they have the band at a 1675 centimeter inverse, which is for the beta conformation of a protein or peptide. So which means that this fiber consists of a beta conformation. So these are just basic studies in order to make sure our compound are well formed. And this here beta conformation is relevant because I said this is an amyloid inspired strategy, right? So amyloid fibers always have a beta conformation, or the amyloid fiber which induce the toxicity have the conformation beta conformation. So I just wanted to make sure my fibers are also behave like an amyloid fiber. So we have seen a band 1675 and 1635, which means that this fiber have an amyloid like a structure. And I have analyzed the emission spectra of this molecule, and this emission spectra is quite important because that can differentiate the self-assembled molecule and without the self-assembly. So this molecule have the CAC 60 micromole. So I have analyzed them above the 60 micromole. So in that case, I can see a peak with a maximum at 450 nanometer. So this belongs to the pyrene hexameric emission, which means that in this fiber, the pyrene could form an hexamer. Maybe I think a hexamer everybody knows if you are a chemistry student, because hexamer, when the two pyrene come close together, they can form the hexamer. So a hexamer usually have a sky blue emission like this. However, below the 60 micromole, I have also analyzed their spectra. They are only in the molecule state, have a pale blue emission like this. So I can now differentiate in the test tube below the CAC and above the CAC by emission. So I will use this strategy in order to visualize inside the cell also. So I can, if it form an hexamer, I can somehow say that my molecule can form the fiber inside the cell also. So for that, first I have seen whether it works. It can go inside the cell and inside the mitochondria. So we have visualized them by a microscope called LSM 780 microscope. This is the first step when we start a research with the cells. So we can culture the cell ourselves in a small plate, cell line, cancer cell line in a small plate, and we will incubate the molecule with the cell, and we can observe them under the microscope. So this is the microscopic image of a cell. We use here a HeLa cell line. It is the widely used model for the cancer cell. So here's a line. Here I can see under the microscope the blue emission of my molecule and the mito tracker. There are all these biological experiments are quite easy. It is not difficult as you see. It is only difficult for the explanation. If you are doing a biological experiment, everything will come easier, because there is a protocol for every biological experiment, and there is an available dye for every biological experiment. We just need to follow the protocol, and we just need to know the theory, how it works, and we should have a little bit experimental skill. So then we can get the data very easily. So I use a mito tracker. This is commercially available. So mito tracker incubate with the HeLa cell and observe under the microscope. I could now say mito tracker means they will only label the mitochondria of the cell. So I could see the mitochondria, and I have incubated my cells with my molecule. So I could see the overlap between the blue fluorescence of my molecule and red fluorescence of my mito tracker, which means that they are perfectly overlap, and which means that my molecule localized inside the mitochondria of the cell. So this is the initial preliminary examination to know whether my molecule is going inside the mitochondria or not. Next, I said I will use this hexameric strategy in order to visualize inside the cell. So hexamer means there is a fiber. Monomeric emission means there is no fiber, there is molecule. So this, if you see the emission range, the monomeric emission is about 4 until 410 or something, and hexameric emission start from 420 to 550 here. This is the hexameric region. Okay, so I have changed the filter of my microscope and then observed at different filter from 380 to 420 and 420 to 480. So here inside the cell, we could see a monomeric emission and the hexameric emission clearly, which means that this is not, but however, this is not a proof that my molecule formed the fiber inside. I can just say they form the hexamer inside the cell. So it can be just, it can also be due to some kind of aggregation. Then also, in order to form the hexamer, people can ask why not the pyrene are just come closer and make the hexamer. That is true. But however, this one just to say that, this one says that my molecule could form the hexameric emission inside the cell also, as evidenced by the emission from the filter 420 to 480. And now I have to give more and more proof to show that the molecule can form the fiber inside the cell. So first we have used a dye called NBD. NBD means 4-nitro-2-1-3 benzoxadiazole, this molecule. So this dye is a special molecule which have a high sensitivity towards its environment. Okay, they can be fluorescent. They can show green fluorescence only when they are in the fibrous state or when they are in the hydrophobic pocket. If they are in the molecule, if they are in the molecular state, they don't have any fluorescent. So what I have done is that I have co-assembled my molecule with this molecule, MitoFF and the MitoFF NPD, co-assembled and observed their structure. So without the MitoFF, this NPD shows only the kind of spear-like structure and they don't have any fluorescence. However, when they are combined with the MitoFF, they have a bright fluorescence like this and they form a fiber. So I have used this strategy to study inside the cell. Now I have incubated my cells with the two compound, with MitoFF NBD and MitoFF. So what will happen, both of the molecule will go inside the mitochondria and they undergo self-assembly over there. And if the molecule are not forming any assembled structure, then I might not see any fluorescence. However, if they are forming an assembly, I could see a bright fluorescence from the cell according to the theory. So I have observed under the cell, so I could see a bright fluorescence when I incubate this molecule with the MitoFF, which indirectly says that the molecule could form a fibrous structure like this inside the cell also. However, without my molecule, this one don't show any emission. So which is an indirect proof that the molecule could form assembled structure inside the cell. And this is also not a solid proof. I have to show more and more strong proof that so that other scientists can believe that this concept is work. So we have checked the transmission electron microscopy of the mitochondria after treating with our molecule. So we have incubated the mitochondria with the mitochondria with our molecule and done some procedure in order to make them ready to be observed under the TM. So this is a solid proof, and this is a strong evidence that nobody can question now. Everyone will believe. Everyone have to believe because this is a proof under I. So you can see that fiber under the TM. You can see the fiber inside the mitochondria very well, which have the diameter, which is quite similar to what I observe in the test tube. So these are the various images which shows this is the control mitochondria image. Those don't have, this is healthy and don't have any fibrous structure. But after treating with our molecule, they destroy the original structure and then occupy all the mitochondria with the fiber-like structure. So this fiber-like structure is the one which is going to destroy the mitochondria and induce apoptosis so that we can kill the cancer cell. And this one, I don't know, you will get confused or not, but I will just explain. So this image shows the tomography image from the TM. So which means that there in the mitochondria, there can also be some mitochondrial membrane. So people can say that it can be a mitochondrial membrane structure, not your fiber. So for that, what we have done is that we have tilted the ankle in the TM and we observe both the fiber from the perpendicular side and also from the side. So this is the fiber when we look from the above. However, when we tilt the angle, we could see it as a fiber. So mitochondrial membrane is a lamellar structure. We cannot see them like that. So this one can effectively differentiate the fibrous structure and the lamellar structure of the mitochondrial membrane. So this data shows as a perfect proof that our concept really works and MitoFF molecule can form fibrous structure inside the mitochondria. And next is all the cellular experiment. There are various options in order to show the mitochondrial dysfunction. Okay, so there are various probes that is available to measure whether your mitochondria is healthy or they are dysfunctioned. So the aim of our concept is that we are using this MitoFF molecule in order to destroy the mitochondria. So now after treating with the molecule, I have to show that mitochondria is dysfunctioned, not the healthy in the cancer cell. Okay. So this is first I have done with the MitoTracker RedFM. So we have labeled the mitochondria with the mitochondria of this cell, observe without any treatment. There is no my compound, just the HeLa cell, just the cancer cell. Labeled with the mitochondria, I could see them as an elongated shape. This is how without any disruption, the mitochondria looks like under a microscope. After that, I have treated with my molecule. Now, the normal mitochondrial structure is destroyed. It is dysfunctioned. So there is no more elongated mitochondria. They are undergo fragmentation and they completely destroyed, which means that MitoFF destroyed the mitochondria. And these are another studies which make solid proof that which make a more proof that mitochondrial dysfunction. We have used a dye called TMRF. This is tetramethylrhodamine. Okay, this one is a dye which we can measure the mitochondrial membrane depolarization. Okay, I said mitochondrial membrane is a negative charge for the, especially for the cancer cell, it is more negative charge. This dye is a particular dye which can detect the mitochondrial membrane polarization. If the mitochondria have a high negative charge mitochondrial membrane, this dye will show bright fluorescence. However, if there is no membrane potential, this will not show any fluorescence. So mitochondrial dysfunction means they are now destroyed. We destroy the mitochondria with the MitoFF, which means that their membrane potential is also changed. Only in the normal state, the cancer cell have negative membrane potential. If they are destroyed or dysfunction, they don't have a negative membrane potential. In that stage, we say that they are depolarized. There is no negative membrane potential. In that stage, we could not see any fluorescence from this dye. So here you can see from 1 hour and 6 hours, it have a 1 hour, it just started destroying. We could see some mitochondrial, we could see some fluorescence. However, 6 hours, they are completely destroyed and membrane is depolarized and there is no more fluorescence or some pale fluorescence. Okay, and we have done some other analysis also. The first one is reactive oxygen species. Reactive oxygen species means they are highly toxic. Whenever a mitochondria is damaged, it produces more and more reactive oxygen species, which is highly toxic to the cells. That can, if there is an enhanced production of reactive oxygen species, then cell will die. Cell cannot survive anymore. So now the mitochondria is destroyed by the mitotracker we have shown, and TMRM shows mitochondria is depolarized, and this data shows they produce higher ROS or reactive oxygen species in the cell. Okay, DHE is a dye that is used for the ROS detection in the cell, and mitosox is a dye which is using for the ROS detection within the mitochondria. So both the dye shows fluorescence when there is an ROS. So I have incubated this dye. I have incubated my cells with this molecule and labeled with these two dye. So you can see now the red fluorescence. So red fluorescence indicate there is an ROS because this dye is designed in such a way that they can detect the presence of excess ROS. So they are fluorescence. If you are interested, you can just Google this dye. Okay, they are fluorescent only in the presence of ROS. The mitosox is also fluorescent only in the presence of ROS. So they show red fluorescence, which means that there is an ROS production both from the mitochondria and both from the whole cell. This data all together confirm that the MitoFF induces mitochondrial dysfunction efficiently. And next we have checked whether it could cause some release of protein from the mitochondria to the cytoplasm or not. So we have labeled the protein with some non-protocol method, and which is initially without any compound, we could see the protein fluorescence well from the mitochondria. However, after treating with the MitoFF, we could see such a fluorescence is dispersed all over the cell, which means that the proteins are released from the mitochondria to the cytoplasm. So as I said, the mitochondria is a huge container of apoptotic related protein. So if mitochondria destroyed, if they release their protein, which means that they efficiently induce cell death for the cancer cell. Okay, and now we have again showed from the cell that there is a from the TM that mitochondria is destroyed. You could see the mitochondria is destroyed and their image is not clear. You could not see a clear mitochondria, and also you could detect traces of fibers in all over the mitochondria, and that comes from the MitoFF fibrils. So all together confirm that MitoFF forms the fibril inside the mitochondria, and they induce the mitochondrial dysfunction, and they cause the release of protein from the mitochondria to the cell. And next, we have also done the supporting information, and this is how we have to make an efficient paper, because we have to be so strong about our data or about our claim. Otherwise, other scientists will just reject your paper or just reject your concept. We have to provide the data in such a way that nobody will not question you anymore. Okay, so here we have shown we have synthesized so many other peptide. So like usually initially I use MitoFF where phenylalanine is the building unit. Now we have changed the building unit with valine. This is an amino acid valine and cyclohexylalanine, and here is a glycine. Okay, so we have make a control compound. So in that the first two compound shows the fibril like structure. However, the last compound shows the micellar like structure. So this data shows the importance of fibril formation in order to induce the cell death. Here you may be now familiar what a TMRM do. They detect the mitochondrial membrane potential, and ROS detect the reactive oxygen species. DHE detect the reactive oxygen species from the cell, and mitosox detect the reactive oxygen species from the mitochondria. So whenever there is a fiber inside the mitochondria, they depolarize the membrane well, they produce the ROS in the cell, and they produce the ROS in the mitochondria. However, when they form a micellar like a structure, they are not depolarizing the membrane, which means that the micellar structure inside the mitochondria cannot induce the toxicity or cannot dysfunction the mitochondria, or the membrane potential is not changed, which means that micellar structure inside the mitochondria is not active. Only the fiber structure can be active. So make a fibrous assembly inside the cell is important. So they also, this glycine, this one which form the micellar structure, also don't produce any ROS or don't produce any ROS within the cell, also don't produce any ROS in the mitochondria. So this data shows how important a fibrillar structure is for the therapy. And now we have to show this is toxic. Our molecule is toxic towards the cancer cell. So how we can prove this one is that we can culture the cancer cell line in our lab. So there are so many cancer cell line available, commercially available. We can just buy it and we can culture. There are different names according to their origin. HeLa cells means cervix cancer, and MDA MB468, which is the cancer cell line model derived from the breast. This is a breast cancer, and PC3 is a colon cancer, if I remember correctly, and SSC7 is a skin cancer. Okay, so you could see a bunch of cell line like this, and you can buy according to your purpose, and you can culture. And what I have done is that I have cultured all cell and tested the toxicity of my molecule at various concentration. Okay, I have tested all the 4 compounds. The first molecule is MitoFF, and the last shown 3 molecule. 2 is fiber and 1 is micellar forming structure. So I have found that all the molecule could induce the toxicity except GG. The micellar structure induced little bit lower toxicity. However, all other compounds are highly toxic towards the cancer cell. So I hope you can understand this diagram. This one shows the cell viability plot. This means 100% viable. However, when I increase the concentration of my molecule, or when I increase the incubation concentration, then there is the viability is less. Only 20% cells survive here. This is how we interpret this data. Okay, so here 100% viable. When I increase the concentration, cell become die. So there is very less cell which can survive when my compound is incubated, which means that this molecule can efficiently kill the cancer cell. And this one is a confocal imaging, which is proof for the apoptosis. So we use a dye called annexin V, which is a dye which can detect the apoptotic cells. So what they do, when the cell undergo apoptosis, they develop so many apoptotic bodies. Okay, so this annexin V dye can bind with those apoptotic cells and show the fluorescence. So whenever there is a fluorescence after treating with the annexin V in conjugation with a dye, FITC, usually FITC annexin V. So this one can show a green fluorescence, which means that the cell undergo apoptosis. Okay, and this is the flow cytometric data which is showing that apoptosis. So this is a facts. This is flow cytometry, means this is confocal imaging. This one means we quantitatively analyze the fluorescence. Okay, so this one, how we read this one, there is a 4 quadrant. This quadrant means cells are live. This quadrant means cells are in the early apoptosis. This one means cells are in the late apoptosis. This one means necrosis. Necrosis means another mode of cell death. Now, here we are not interested in necrosis. We are only saying about the apoptosis. So, if I have a more population of cells in this quadrant, which means that my cells are undergoing apoptosis after treating with my molecule, and most of them are in the early apoptosis and some are in the late apoptosis. So this whole data shows that my compound can kill the cancer cell efficiently. And here you can see some two data which is not inducing any toxicity. So these are the normal cells. So HEC293 and the NIH3T3. These are the model for the normal cell line. These are not cancer. These four are cancer cells. These four are the normal cells. So I have incubated my compound in the normal cell also. So it is quite good, quite friendly with the normal cell. They don't induce any toxicity, which means that this type of compound, this kind of intra-mitochondrial fibrillation can efficiently differentiate a cancer cell and the normal cell. And why this happens and why they have to differentiate the normal cell and cancer cell. For that, we have checked the concentration of the molecule inside the cell in both HeLa cell and the normal cell. So normal cell, we have used the HEC, it is called HEC293T cells. So when we check the intramitochondrial concentration, we find that the final concentration inside the mitochondria is in the millimolar range for the cancer cell, but in very low for the normal cell. Why that happens? That happens because of the TPP. So I said that mitochondria have a negative membrane potential, and this TPP coincide because of the negative membrane potential of the mitochondria and localize. And this negative membrane potential of the cancer cell is found to be quite higher. That is not my research. That is already known. That is already reported. They have higher negative membrane potential for the cancer cell. But normal cell also have negative membrane for the mitochondria. All the cells have negative membrane for the mitochondria, but amount of negativity is quite higher for the cancer compared to the normal. This is why this kind of molecule can differentiate normal cell and the cancer cell, because they need to accumulate more, and their accumulation will be in accordance with the negative membrane potential. If they have a more negative, then they could go more and more inside in order to overcome their CAC. However, less negative membrane potential means their accumulation inside the cell is also reduced, so that the final concentration is not much higher. So they make a huge and dense fiber inside the cancer cell, but not in the normal cell. And this is why we can differentiate the cancer cell and the normal cell by this method. And this is just an explanation why the mitochondria of the cancer cell have more negative membrane potential, and that happens because some proton gradient across the inner membrane of the mitochondria. And this is another huge area of research. So basically what we have to understand is that cancer cell have a hyperpolarized membrane for the mitochondria but not for the normal cell, so that we can differentiate this kind of TPP molecule can differentiate cancer cell and the normal cell. So next we have proved why this mechanism, why the fiber induced toxicity. So for that we have created a liposome. So this kind of model liposome by using the cholesterol and POPC, like such a model liposome we have created by our hand to mimic the mitochondria. So we are only interested in the membrane. We are only mimicking the mitochondrial membrane. I said mitochondria have a membrane, double membrane, outer membrane and the inner membrane. We just want to mimic those membrane in the test tube in order to analyze, in order to find the mechanism why fibers are toxic when we are targeting the mitochondria. So we create this kind of model liposome and we have incubated with our molecule. So we use liposome that is encapsulated with a dye, calcine dye. So in the normally there is a dye inside the liposome. We could see very less fluorescent, and now we have incubated with our fiber, with our MitoFF fiber, and we could see a release of fiber from release of dye from the liposome to the outside, which means that the fiber destroy this liposomal membrane and release the dye from the from inside to the outside. Indirectly, this says that the fiber destroy the membrane. So this could be happen in the mitochondria also. The fibers are formed inside the mitochondria, and this fiber might be destroying the mitochondrial membrane. Okay, we have analyzed the release of the dye by the if I remember, by the fluorescence microscopy, fluorimeter, by the fluorimeter. So we have analyzed the dye release. We have detected the release of the calcium from the liposome to the outside in a time-dependent manner. So Mito FF, which form the fiber, destroy this membrane and induce the leakage of the dye, but not the GG. GG is, if you remember, which is the micellar forming one. So micellar is not interacting with the membrane, but only the fiber is interacting. And now we have experimentally proved that our concept really work. And now we have to also give from theoretical explanation whether this one, how this one works, and some kind of calculation or something. So what we have done, we have conducted a molecular simulation analysis. So we have made a liposome by the simulation. We have made a model liposome to mimic the mitochondria, and we have made our structure by theoretically we have created our Mito FF fiber structure, and we have done the interaction study between this kind of liposomal structure and the fibrous structure, and study how they interact theoretically. We have proved that, we have seen that this kind of fiber can effectively penetrate the mitochondrial membrane well and induce a pore. Okay, and eventually this will destroy the mitochondrial membrane, and that is why we could release the apoptotic bodies or the related proteins from the mitochondria to the cytoplasm that will eventually cause the apoptosis. But when there is a micellar structure interacting with the membrane, there is no result. They are not penetrating, they just stay in the surface, which means that the micellar is not effective to destroy the membrane of the mitochondria, but fiber can do it well. It can efficiently penetrate to induce a pore. And after that, after we have done all the in vitro analysis, and after we have done all such a theoretical analysis, we have checked the in vivo. This is the first step towards the clinical trial. We have to experiment them in an animal. We usually use the mouse model. So there are so many other models. We usually use the mouse model. We have made a tumor initially in a mouse, and then you can see this is the control. How this is do, we can I said there are so many cell lines which is cancer cell line, so we will inject those cancer cell line into the mouse and develop artificially a tumor in the mouse. And this one we have allowed them to grow, and we have treated with our peptide, the Mito FF peptide. So here Mito FF we have injected. So this control shows without any injection of the compound. This diagram, the bottom line shows after injecting our compound. So you could see how the tumor size is differed from the control. Without any treatment, the tumor continued to grow. But after we inject our molecule, the tumor is reduced, or almost no tumor. So which means that they can efficiently inhibit or destroy the tumor. So this is the quantitative analysis which is showing that this is the control have a huge tumor volume. However, after treating with our molecule, tumor volume is quite less, which means that this can efficiently reduce the tumor. And this one shows the body weight analysis. Like we have to also check whether the molecule can induce toxicity towards the mouse, or mouse are alive. So after the 14th day, after the treatment with our molecule, mouse is still healthy. They don't have any change. They don't induce any systemic toxicity. So mouth is healthy, which means that the molecule no toxicity in the normal condition. However, they just reduce the tumor. So now we are on the way to develop this one towards the clinical trial or for the practical application, but still we have to overcome those many barriers in order to make. But there is a huge possibility, or the chances for this one to be successful is quite high, because there are with the available method, there are many drawbacks. For example, for the conventional drug, they induce fatal toxicity and side effect Fetal toxicity means toxicity for the normal system. However, their advantages are quite less. Okay, but the protein inhibiting drug, they cause resistance, or they are quite complicated. They have a very complicated synthetic step, and the one thing is that they also have an advantage that they can be highly selective to the tumor also. But the protein inhibitor cause the resistance, which is a big problem. If they reduce, if the drug cause resistance, then it is not useful in the end. In the end, we will again have a tumor. So compared to all other available strategy, this kind of mitochondria targeted drug-free agent, we given the name drug-free because we are not using any conventional drug. We are using a new molecule which induce the toxicity by nanostructure. So if we have this kind of structure, they have a limited barrier for the application. However, they have a lot of advantages, such as they work under low concentration, that is quite easy to produce. They have the high selectivity towards the tumor. Okay, so so until now, all the data I have proved that the MitoFF kind of molecule, or the molecule which can form the assembly inside the cell, can cure the cancer well, and they can also reduce the tumor in the real in vivo condition in the mouse also. So in conclusion, we have introduced for the first time an intramitochondrial self-assembling peptide amphiphile called MitoFF, which showed an enhanced accumulation inside the cancer cell, and it induced the mitochondrial dysfunction efficiently, and it is showed efficient cancer killing activity both in vivo and in vitro. So at present, we are also modifying this one. So now we have used only the molecule. We use MitoFF as a molecule. So there are certain barriers in order to use molecule in the in vivo application or the practical application, because small molecule can be cleared from the body very well. So now we are making self-assembled structure in the outside in the kind of micelle, and which will be responsive near the tumor cell and disassembled and then efficiently uptake to the tumor cell. This kind of strategy is more better because they have long circulation time in the body. So this might be the end of my presentation. So I thank my professor for helping me with all this research, and we have a collaboration in different part of Korea, like a KIST or just other universities, and I have so many collaborative professors within the UNIST and other universities in Korea. And I thank my lab member, and I thank everyone for being here to be so patiently to listen my talk. And I thank Dr. Vijay for inviting me for this talk, because it was quite surprising when he called me for this talk, but I was so excited to present my work in front of Kerala. This is the first time I present my work in Kerala, and also he is just like my brother, so I thank him very well for inviting me. So thank you for your attention, and please ask your questions now. I hope it's clear.
Yes, ma'am. Jeena, ma'am, since we're nearing the end of the session, I mean, we're past the time for ending the session, I think we'll just take one question before we wind up, if that's all right with everybody. There's a question over here by Mini Krishna, ma'am. What is the difference between L-Mito and D-Mito in vivo tumor reduction analysis?
Okay. Actually, L-Mito is a naturally occurring peptide. Here we have used the L-peptide. All what I explained is L-peptide. So L-peptide is naturally occurring peptide. So the problem with the L-peptide is that they can undergo a reduction, a biodegradation, because they are naturally existing. They can be degraded by our body, okay, because of so many digestive enzymes. So we think that the D-peptide will be much more better because they have a high resistance towards the proteolytic enzyme. So, but in case of D-peptide, normally the D-peptide are more efficient for the therapy compared to the L-peptide. However, when we do with MitoFF, that both the D and L showed the same behavior, which means that they behave little bit differently from the real scenario. That happens because they make an assembly inside of this cell. So basically, the answer for your question is, in here, L and D does not show much difference. But normally that is not the case. If you use a fiber like fiber for the therapy, they will show more efficiency because they have high resistance towards the proteolytic enzyme. So I hope the answer convinces her.
I believe so, yes, ma'am. No more questions on the chat box, and I'm sure that they have noted down your email ID, so they would be corresponding with you, I believe. Oh, yes, Mini Krishna ma'am has responded. She said very informative and organized, yes. So Dr. Minimol has posted the feedback form link on the chat box. So do drop in your feedback, and you'll be receiving, the participants will be receiving the certificate shortly. We come to the end of the session, and on behalf of the IQAC and the Research and Consultancy Cell of Vimala College, Thrissur, Kerala, I express and place on record my deep sense of gratitude and appreciation to you, Dr. Jeena, for spending this time and for spending your intellectual resources to such a great and useful extent with the entire participating crowd. From the presentation and from the way you so humbly and so clearly presented your topic, we can see the dedication that you have towards research. It is not only the knowledge that you have shared that we'll be carrying homes or we'll be carrying with us throughout. We'll also be, we've observed the kind of meticulous detail that has, and the intense study that has gone into your research. So we are going to take motivation from all of those, and of course, all the very best to you. Thank you for accepting our invitation and being a part of our humble venture, EXPLORIGATE. So a big thank you, Dr. Jeena, and all the very best for a wonderful future, ma'am. I also place on record my deep sense of appreciation on behalf of the IQAC and Research and Consultancy Cell to our respected principal, Dr. Sister Beena Jose, a great academician, a scholarly person who is an inspiration in every sense for research and academic ventures. A huge thank you to Dr. Mini, IQAC coordinator, our strength and the brain behind such wonderful and industrious ventures. Of course, thanks is also due to all the IQAC co-committee members and all the members of the Research and Consultancy Cell, and congratulations to the Department of Chemistry. A word of appreciation to Dr. Bijoy's efforts as well. And of course, participants, we hope that EXPLORIGATE has been a significant part of your research endeavor, and we hope that this day and this session will be a significant part of your journey in academics. Wishing all of you a great day ahead. Thank you from Vimala College.
Thank you, Maya.
Thank you, Maya.